Christopher. Meow.
Okay.
Breakfast. Alright, that's enough.
So we need to talk about selective permeability.
Oh, wait.
You're going to allow some materials to cross, but not all materials. Okay, now I'm going to draw what I would have drawn over here up here because I have to, so Chris can see it. But we had that generic membrane we were talking about yesterday.
Where do we draw?
This doesn't necessarily have to be in your notes, but if you want to, just use your margin. Alright, so remember there's two tails per, but I'm entirely too lazy to draw all of those. But some things need to be able to cross the membrane. So you have certain things you need, like oxygen. You want that to get into your cells. Because oxygen is small and nonpolar, that's one of the next little notes under what we just wrote for definition. Since oxygen is small and nonpolar, it can cross directly through the membrane without any problem. Remember that inside where those hydrophobic tails are, it's very nonpolar in here. So if the molecule is going to be able to cross directly, it needs to be a nonpolar molecule. Oxygen that's dissolved in your blood and ultimately carried to your cells needs to cross those membranes easily so that you can use it. You also produce stuff in your cells that you need to get rid of. Carbon dioxide, ultimately you're making it as you respire. That concentration gets bigger and bigger in the cell. You got to get rid of it because it's toxic. It can pass directly through the membrane because it is small and nonpolar. So it's really easy for that transport of oxygen and carbon dioxide. They move across the membrane directly. They don't need any help. But then we also talked about those channel proteins, right? Some things are not small and nonpolar. Maybe it's small, but it's polar like water is. You still need water to be able to go in and out of your cells. So they need those little openings, those channel proteins to pass through, generally speaking. So if anything's polar, if it has a charge, it can't go through the nonpolar part of the membrane directly. So it has to go through a channel protein, but generally speaking, you need certain things to go into your cell. If they're small and nonpolar, that's not a problem. Oxygen can go in and you can get rid of carbon dioxide really easily. Water can pass through those channel proteins. But if you have large molecules, they tend to be moved through vesicles, and we'll talk about that in section 5, so we don't really have to worry about it yet. But small nonpolar molecules cross the membrane directly by themselves, no problem. Small polar molecules can be carried across by proteins. If you have those channel proteins, they can move them across. Some of those proteins don't just let them pass, they force them to go in a certain direction. So we'll talk about that in the next section, which will actually start today. But you have a whiteboard question here. You're supposed to try and think of another semi-permeable object. A membrane is semi-selectively permeable because it allows some things to go through, but other things not. So think of an everyday object that allows some things to go through, other things not. And just write it down on your whiteboard.
It shows an object.
Everyday, yeah, just some object that allows certain things to go through, other things not. You might use something like this maybe when you're fishing. Let some things through, other things not. Maybe you're just making spaghetti.
Oh, good.
And you're going to let some things through, other things not. maybe
What's the thing called?
there are examples in this room that you can think about. Let some things through, other things not.
Got
That would be a good example.
Wow, cheater.
Used to know.
That's a good example. It's a little outside of the box, but it works. That would be a good example. Yes. Boom. Yes.
Oil.
So for me, it's a filter for stock.
Yes.
Yeah.
Yes. Yes.
I have a question. Oil.
Yeah. So you take the particles out, and the oil goes through. So that would be a good example. So I got lots of different examples so far. I liked pretty much all of them. The only real thing that you gotta make sure you do to get all points is tell me what goes through and what doesn't. So, for one example, Sarah, what'd you have?
The windows.
The windows. What goes through the windows?
I don't know.
Sunlight does, but air
I was
doesn't.
thinking a screen.
I know
That
what
is a
you
good one.
So the screen would be your example if you want to use the screen. Air can go through, bugs can't go through. Window is a fairly outside the box thought because the thing going through is light. And you don't usually think about that. And you're thinking about objects traveling through, not necessarily waves of energy. But you could use the doorway if you wanted to. If you were talking about me versus an elephant, I can go through, but an elephant can't.
Hey, Corbin.
Find a seat, please, and get your stuff out and get ready to go. Okay, so that was a good example. Window is, screen is. Anybody have anything different?
Fishing net.
Fishing net, water goes through, the fish doesn't. Yours?
Oil filter.
The oil filter, the oil goes through, the filter takes out particulates, bad things. A water filter does the same thing. I don't know.
Air
Comment
filter.
out there.
Soil.
Air filters. Soil filters stuff out of the water so that that aquifer's nice, clean, fresh water with nothing in it that's bad. All those things would count.
Water filters everything except things above the density.
Okay, receptors. You need to fill in definition here probably. And you're looking at page 80 for a picture here, and we're going to try and draw these pictures in your notes, but you might have to look at theirs to see a better version of the same thing if I do a bad job of drawing it. I want you to understand this basic idea.
What page is it on?
Page 80. So receptors are proteins that detect a signal and act because of it. The terminology you're going to use for this, ligands are the signal molecules, so you might connect those things, either underline them or whatever. So the signal is a ligand that's going to attach itself to that receptor that's an inactive protein that isn't actually doing anything currently to make it an active protein that can do whatever function you want it to do. And there's two kinds of receptors. There's intracellular ones and there are membrane receptors. So you look at the two pictures on page 80, and we're going to draw those two pictures, so make sure you have a little bit of space for this. I'm not going to draw the membrane the way they did. I'm going to simplify it and just make it two lines. It's a bilayer still, as long as I label it and say, hey, this is my membrane. If I could spell, then it's not going to be a problem. I'll know that that's what that's supposed to represent. So that's my boundary. And intracellular, when I say that intra, all I'm saying is that that receptor is inside the cell. So if I have out versus in labeled, the receptor's in here, and I'm going to draw it with a certain shape with a little cutout like we normally do. the ligand is over here, and it's a shape that fits into that cutout. So you might want to make sure everything's kind of labeled, right? I was already using green for ligand, so that's this here. My receptor is a protein, but right now that protein's inactive. It's not functional. It's not doing anything. There's a vent that the wind's pulling up and down over there. Sorry, I can't control that. So anyway, you got ligand, you got a receptor. What's the problem?
There's something in between them.
There's something in between. So in order for this ligand to get to the receptor, I have to do what with it?
Go through the membrane.
Go through the membrane. Can all things go through the membrane directly?
No.
No. So that creates a problem in this context. But if this ligand can get to that receptor, it should attach itself to that receptor and activate that receptor. So I'm going to change its shape. All right, so I went from here to here. And now I have this active protein. I wanted to highlight, not. That can do whatever it is that it's supposed to do. So we drew something similar to this before. But that was when we had a substrate that changed shape. In this case, I want that receptor to change shape because that's the thing that becomes active when before it was inactive.
So in order for it to become active, it has to change.
So I need this receptor to attach to it so it changes, so it becomes active. Exactly. You're right. But talk about the ligand too. You need that to make it change. Okay, so that's for my intracellular. I can also talk about the membrane receptors, and they're a little bit different. So I'm going to draw pretty much the same picture. I'm just going to put my receptor in my membrane instead of putting it inside the cell. So I had a problem before because the ligand needed to do what to get to the receptor over here?
Go through the membrane.
But does it still have to do that?
No.
No. So now I don't have that issue. The ligand can get to the receptor without going through the membrane. If it does combine with that receptor is supposed to change, so I need to still have it in the membrane, so the change version's here with the receptor. But as you can see, part of that activated protein is still inside the cell. So maybe it can still catalyze reactions inside the cell, right? Unlike this one over here, this enzyme would be free to go and find the reactants. The reactants would have to come to this in the membrane. But I still could activate that protein without making the ligand go into the cell. So there's a pretty big difference there. All right, now if I'm going to ask a question about this on the test, and you might want to pay attention because this is probably the question I would ask on the test. You would want to have a good answer to it. And it's not a hard question because I basically just told you the answer. So I'm going to pick on somebody. You got to think about it for a couple seconds. But which kind of receptor, intracellular or membrane receptor, is more difficult to activate and why? So think about that for a second. That's all I'm going to ask. So if you have a good answer, you can start thinking about writing it down.
Yeah, what's the question?
Between that intracellular receptor, this one here, and the membrane receptor, which one is harder to activate and why? So I'm going to ask somebody, but then you might want to write a good answer.
Someone grab tissues for Corbin. Corbin. Right here. Here's right here.
That one will work. Okay, so hopefully I get a good answer here. I gave you a little bit of time. Which one's harder to activate and why, Berkeley?
I would say intracellular because it has to pass through the membrane.
Because what's it?
The the ligand
The
has
ligand.
to pass through the membrane.
Has to go through the membrane. That's good.
Which is harder to activate.
So this one's harder to activate because you need the ligand to enter the cell before that receptor can be activated. So that makes the intracellular receptor harder to activate. Not all things can pass through the membrane easily, so that's the issue. Or I could just flip the switch on you. On the test, I might say, which one's easier to activate and why? So you still have to talk about how the ligand may or may not have to go through the membrane. You just have to reverse everything that you're saying. You'd say that the membrane receptor is easier to activate because the ligand does not have to pass through the membrane. So I could switch it up still, but I'm going to ask you a question, something like that. So make sure you have an answer. Okay, again, at the end of the day,
Remember me?
if you're drawing pictures, make sure they're labeled well so that you understand what's going on. So otherwise, you'll look at this later and you'll be like, I have no idea what that is. So I labeled with red the receptors. Right, the activated version is with the orange. The ligands are green. Just make sure you have a good label. You can just put a little L next to this and an R next to this and AP next to that for activated protein. Whatever. Okay, so we're actually done with those notes. And we have to start some notes for section 4.
Days, no days, days, no
I know you guys are probably not interested in starting new notes, but
I'm very
we're gonna have to use our time productively. Because you guys are already done with the section 3 work, you can't just sit here while you don't.
I haven't even started yet. I haven't even started. What are you talking about? Dog.
I got it done.
How many people are done reserving? I don't know if
But even if you're not done, that's really because you didn't do it, not because
No, it's because we got it at the end of class.
everybody gets some notes.
I got it done.
Yeah.
Okay, so we're not gonna talk about anything too mind-blowing from here on out in class, but you still do need to pay attention. It can be difficult if you're not gonna pay attention and you're not gonna follow along step by step. This section starts on page 81, so you got your book open still, you might as well just flip the page and get there. It's on diffusion and osmosis. Our objectives are gonna be to talk about passive transport and describing that. Also, to go with that, we're gonna talk about active transport, but we'll get to that in the next section, I believe. And then we're going to distinguish between osmosis, diffusion, facilitated transport, all of which can be passive depending on the context. Right, I just talked to you guys not too long ago about how some things can go through the membrane directly, and they'll do so automatically. And some things need a little bit of help, like the water, to go through the channel protein. But at the end of the day, none of those transports that we talked about on this slide require energy. It just happens automatically. So they're all forms of diffusion. They're all passive transport. So where are we? Way over here. Okay, so we need to know what passive transport is. It's movement of molecules that does not require energy. I'll show you guys a demo. Chris is gonna miss out on this, but I can show it to him really easily some other time.
What is that?
I got water here, just water. But there's molecules in this water, and those molecules are always moving. You guys know that. Even if you can't see them directly, I mean, you see water when it's moving. If it's boiling, when they have lots of energy, you can definitely tell that they're moving. But when it's cold water, you might not necessarily notice that they're moving, but they are. Right? Even the particles in this desk, it's a solid, but they're still moving. They're vibrating around fixed positions, but the only time particles stop is when that temperature reaches absolute zero And that doesn't really even exist. It's just theorized. So particles are always in motion. And if you got a cell that's full of water, right, those particles are moving. It's not solid, so they're not just stuck in fixed positions, still vibrating. They're definitely allowed to move and flow, and everything is moving and flowing around in there, even if you don't notice it. So we're going to talk about movement of molecules that doesn't require energy. We're going to talk about concentration gradients. A difference in concentration from one area to the next. So I'm going to put some food coloring in this water. So I'm going to put a drop of this yellow. I'm going to put a drop of blue. And you can see right now where that dye is really concentrated. Because it's all just in one little location, but eventually that dye is going to spread out and you're going to have an equal concentration throughout the whole solution. So we have a concentration gradient that you can see right now. There's yellow, there's blue, and then there's just clear because the yellow and blue hasn't gotten there yet. So there are different concentrations in that solution we're looking at. So there should be some kind of diffusion, ultimately. Molecules always tend to move from areas of high concentration to areas of low concentration. Another demo that I could think about doing, but you guys would probably all get injured in the process, and if I shoved you all in this corner over here and told you to start moving randomly, just start moving in one direction or another, and if you run into something, turn around and move the other way, eventually you guys would all spread out throughout the room. Like I said, I can't really do that because you'd all end up injured or mad or whatever. But the basic idea is pretty easy to follow. If you're all stuck in one place and you start moving, you have to spread out. It's impossible for you to all stay in that same place. So the same thing in here. You have areas of high concentration and low concentration, and things are going to start to move to equilibrium. Right? They're trying to spread out, those molecules are. So I'm not even going to touch this throughout class today, but eventually yellow and blue make what?
Green.
So you're going to see that the entire solution is not going to look blue and yellow, it's going to look green, and it's going to happen automatically because particles are moving. Even if they're moving slowly, they're still moving. I could make this happen faster, but what would I have to do?
Heat it up.
I'd have to shake it, stir it, boil it. I could heat it up,
What
but
if you
all of
freeze
those
it?
things require energy.
It would take slower if you cool it down.
And we're talking about passive transport. That doesn't require energy. So I'm just going to leave it alone. It should do it anyway. It should move and mix anyway without any extra energy. It might happen a lot faster if I put energy into it, but it doesn't require energy. That's what diffusion is. So you're always going to have things moving down or with their concentration gradient. When I say that, sometimes you just like, what does that mean? Right? When I'm saying with its concentration gradient, or if I say down the concentration gradient, I'm just saying that things always move from areas of high concentration to areas of low concentration. So it's going to move from where the blue is now to where it's not, which is the rest of the water, until you can't even tell there's blue anymore. It's all just green because it mixes with the yellow. And it happens automatically. It doesn't require any energy, and that's important. If it costs you energy to get oxygen inside of your cells, that'd be a problem. You needed the oxygen to get energy. So without energy, how are you going to get oxygen in your cells? It wouldn't happen. But it works because the concentration of oxygen in your blood is supposed to be a lot higher than the concentration of oxygen in the cell. Because you're constantly using that oxygen in the cell and it's turning into water in that respiration process. Whereas you're breathing as we speak, hopefully, and you're replacing that oxygen in your blood, and there's a constant amount of oxygen in your blood so that you can get that into your cells. And this transport is simple diffusion. It doesn't require energy. It happens on its own. If it cost you energy, it wouldn't be worth doing. You wouldn't have the energy to make it happen because you need the oxygen for that. Carbon dioxide, there's a buildup of that in your cells because you produce it in respiration. It's toxic. You need to get rid of it. But again, as long as Jake's still breathing, that carbon dioxide concentration in his blood should be relatively low compared to that of the cell, because once that blood gets to the lungs, the carbon dioxide leaves the blood, goes into the air that you're breathing out right now. So that needs to happen automatically, and it does. Now, if Jake stops breathing, then this concentration starts getting bigger and bigger. And then, are you still going to have carbon dioxide exiting your cells? Not as quickly, and maybe not at all. And then you're going to have issues. Also, if Jake stops breathing, I'm probably not going to have a constant amount of oxygen in my blood. That's going to continue to drop until you have nothing left for your cells to use. So you'd have some problems, some issues. But things always move from high to low. So these next two questions, they're really kind of demo, well, not, I'm going to put drawings up here and we're going to go through the questions together. You still got a whiteboard out, right?
Yeah.
We're going to use that. So I have two solutions separated by a semi-permeable membrane.
Is this gonna be in our paper or no?
Yeah, put the picture just like I'm putting it. And you're not going to answer this with words, you're going to draw. Hopefully you understand the drawing. I have solution A and I have solution B separated by a semi-permeable membrane. It says solution A has a higher concentration of salt than solution B. So salt is made of sodium and chloride ions. So I have lots of ions floating around on this side because it's got a high concentration. Doesn't really matter how I put these in, I'm just trying to kind of do it randomly. So lots of little ions floating around, it's very concentrated. Whereas solution B has a very low concentration. There's one sodium ion and there's one chloride ion. That's it. So I told you that things just move randomly. It's not like they actually choose where they're going, they just kind of move in random directions and they tend to move away from each other, ultimately. But this sodium ion here could move across the membrane from B to A. And this sodium ion here could move across that semi-permeable membrane from A to B. But there's a tendency in this scenario for it to move more often in one direction than the other. I told you things always go from high concentration to low concentration. So on your whiteboard real quick, don't blurt out an answer. You're either writing left or right. Which direction net, even though sodium can move this way and that way, overall, which way is it going to move more if I just let this solution work its way to equilibrium? So it's just left or right. What direction does the salt want to move?
I know. I was just thinking directions now. Let's go.
Okay, so I have a lot of answers, and overwhelmingly, you're telling me that it's moving in this direction net,
But it's not.
which means that more sodium ions are going to make their way this way than are going to make their way back. Remember, it is random motion, so technically some will move to the left, but more will make their way to the right than to the left. So overall, that's my answer, that red arrow. It's going to move to the right. So if you need to write that out somewhere else, if you drew all the other arrows, then you're going to be confused. But generally speaking, solute always wants to move from high concentration to low concentration. But that red arrow, is for the solute and wants to move to the right towards solution B. Okay, everybody good with that?
Is there the dog on top of school?
Now we got the next question. We have the same solution, two solutions, but one slightly different factor. I still have a high concentration of sodium chloride in solution A and a low concentration in solution B, but this time we're assuming that sodium chloride, the sodium ions and chloride ions, can't move across the membrane.
Oh.
Because maybe there's no channel proteins for them to go through, whatever, doesn't matter. But if this salt can't move, but the solvent can, the water can, you also have to know which direction it's going to want to move. So things always move from high concentration to low concentration. But you have to think about for a second, and this is going to be another whiteboard thing just to make sure that we all kind of get this and we're all on the same page. If I have a high concentration of salt over here and I have a low concentration of salt over there, which solution has a higher concentration of water relative to the salt? They probably have the same amount of water. It's the same volume on both sides. It doesn't matter. But which side has a higher concentration of water relative to the salt? So this time you're writing A or B, or you can probably still do left or right. So which side has the higher concentration of water relative to the salt? This one I would guess that maybe we might have a few more wrong answers, but that's okay. Which side has a higher concentration of water relative to the salt? So still left or right or A or B, it doesn't really matter how you answer that.
Scared me.
Behind your head, that's it.
Zero goes.
Okay, actually most of us still doing a pretty good job. If this is a low concentration of salt, that means relative to the salt, I have a fairly high concentration of water. And this would be relatively low compared to the salt. So now again, on your whiteboard, it's either left or right. Which direction does the water want to move? If the salt can't move and the water is going to move instead, which direction is that water going to move? The left or right answer. Remember, water is actually going to move in both directions, but more of it's going to move one way than the other because it always wants to move the way everything always wants to diffuse. Okay, we're doing a good job. We are so smart. I told you this stuff wasn't hard. You just have to make sure you keep up step by step. The solute wants to go this way. But if it can't move, and instead the water has to move to make this kind of even out, the water is going to have a net movement to the left because that's where the low concentration of water is. So when we have this beaker here still with the colors that are mixing right now,
It's green.
not only is the yellow and the blue starting to move to areas of low concentration, but you also have water from the low concentration areas that's moving in between those particles and helping pushing them apart. Everything's happening at once. All the molecules are moving. But water tends to move towards high concentration of solute. And we usually refer to the concentration of the solute, not the water. So you have to think about that relative. If it's high concentration of solute, that actually would mean a low concentration of solvent relative to the other thing. So that's when we start talking about osmosis.
Diffusion of water.
It is the diffusion of water. So fill in your blank, whatever needs to be filled in. And on this one, you guys might want to make a correction because I screwed up. This was not supposed to say low to high. It was supposed to say high to low. So you might want to fix that. My brain was tricking me because I was still trying to think about going towards high concentration of solute. So although the water does want to move from high concentration to low concentration from its own perspective, it still does move towards the high concentration of the solute, and that's what we usually talk about, is the concentration of the solute, not the water. So you have to think backwards a little bit when you're doing that. Now before you flip it to the other side too, there's a certain amount of pressure associated with osmosis. If I had a tube that had kind of a U-bend to it, and I put water in there, the water level would be the same on both sides, right? Gravity would make it be the same on both sides. But if I put a semi-permeable membrane down here, and I make it so that there's a low concentration of salt on one side versus a high concentration of salt on the other side, and that salt can't go through that membrane, but the water can move. This is osmosis again. So, one more time. If I put low over here and high over here, concentration of salt, which direction does the water want to move?
Low. No, low to high for this water.
Okay,
Well,
I
there's
just
higher
want you to write it.
concentration of water over
Write
here.
it on your whiteboard, left or right. Which direction is the water going to want to move here?
Oh, in there?
Yeah, the salt can't move. I didn't put the concentrations of the water in there, but you can infer what they would be. Which direction does the water want to move? To the left or to the right? You've got different answers. I'll talk about it.
Because,
Oh,
yeah, because,
yeah.
because there's a higher concentration of the salt on that side.
I
Doesn't mean it's right, but
I knew this, but I
you're the different answers.
Mr. Morris, am I right?
Right.
You guys have different answers.
What?
I knew which I knew.
What we're gonna do. Okay, so somebody's gonna be able to give me the right answer. And even if not, we'll figure out the right answer, because if you're wrong, then the other answer's right. But which direction do you think the water wants to move, Jared?
Right.
And you would be correct.
Because there's a more concentration, so there's a.
The water's gonna move towards the high concentration of the salt because that's where the low concentration of the water is. But if the water moves to the right, what does that do to this water level?
Goes up.
Look what I just did.
And that's working against gravity, correct? So and that actually will happen if you set up something like this because there's a certain amount of pressure that osmosis can put on this situation. Whatever pressure, whatever force you'd have to use to push this side down and make it balanced again, that's equal to the osmotic pressure. And if you think about that in context of a cell of that little water balloon, what happens when that pressure builds up too much? It pops, and your cells literally can pop if they're in the wrong conditions. Because if there's a big pressure into the cell,
Does that hurt?
your membranes you wouldn't feel it necessarily, but if somebody gave you the wrong kind of IV solution, it would kill you ultimately because all of your blood cells would pop.
I've got a question for you,
It's
Mr.
not okay.
I've got a question for the teacher.
Yeah.
Okay, so osmosis, what's it supposed to do to water? Purify it or what?
Oh, when they do the filtration by osmosis?
Yeah. Like Gush does it, but